So terrific. Thank you so much. It's really a delight for me to be back at this amazing meeting with the beautiful weather and lots of friendly faces here in the audience. And I'm looking forward to telling you over the course of the next couple of minutes about the progress we've made at Semma over the last year since we last spoke. So just to remind you who we are, our mission as a regenerative medicine company is to make curative cell therapy a reality for diabetes. The company was founded in 2015 based on work out of Professor Doug Melton's laboratory at the Harvard Stem Cell Institute. And for those of you who know Professor Melton, he has 2 children with type 1 diabetes and has dedicated his career to making a new kind of therapy possible in type 1 diabetes. And Semma is named after those two children, Sam and Emma. So I'll tell you a little bit over the course of this presentation about how we see the path to making that curative cell therapy a possibility. And our company as a whole is really very much laser focused on disease areas where there is significant unmet clinical need, and combining 2 of our innovation engines within the organization to apply to those areas of unmet need. The first being breakthrough cell technologies, and we leverage a lot of what's been learned in the pluripotent stem cell field to get there, and I'll show you a little bit of the updated data on that, as well as an innovation engine on the delivery side. And we just heard in the last talk how important delivery is for cell therapies, and that's absolutely true in the case of cell therapies for diabetes. And then mention at the end a little bit about our path to clinic and the team that we've been able to build in terms of both people operating in the company and also investors over the last year. So I'll start just to remind you that the idea of curative cell therapies is not new, and in fact we've had several decades of remarkable clinical experience with bone marrow transplant, which is the original of the curative cell therapies that continues to this day. And that's been augmented and built upon in really remarkable ways by many of the people here in the audience and at this conference. We saw, of course, the CAR-T approvals in the last couple of years. And of particular interest to us, this extends into the diabetes field as well, where there's been really quite remarkable progress clinically with organ donor-based islet transplants, the islets being the part of the pancreas that contains the insulin-producing beta cells. And what we hope is to leverage all of this work that's been done and to stand on the shoulders of these giants in the cell therapy field and move stem cell-derived islets forward as the next big breakthrough here in curative cell therapy. So to remind you of the true unmet need in this space, diabetes is something that affects, I think, virtually all of us, whether it's a family member, friend, or neighbor, and it affects all parts of the world. And more than 50 million people really require insulin injections worldwide, meaning they have an insufficient beta cell mass to control their metabolism. And so that's an amazing population for which new therapies are desperately needed. So to remind you of the 2 types of diabetes that make up the majority of that pool of people, there's type 1, which is a disease you can see on the bottom panel here, an autoimmune disease where the insulin-producing beta cells that normally reside in the islet of the pancreas are destroyed by the patient's own immune system, and that leads to complete loss of insulin production by the patient's endogenous pancreas. And then type 2 diabetes, which starts differently, has a different etiology, but in severe cases ends in exactly the same place, where we have insufficient beta cell mass to control the patient's blood sugars. And about half of the insulin sold and injected into people is injected into patients with type 2 diabetes. So we think there's significant opportunities in that space as well. So I alluded to in the very first slide in that history of curative cell therapies that diabetes can actually be cured, type 1 diabetes can actually be cured with an islet transplant. So over the course of the last 20 years, hundreds of patients have been treated with islets isolated from organ donor pancreas, where they're put back into the patient's body under immunosuppression. And we had the remarkable opportunity of having one of these patients come and speak to our scientific team just a couple of weeks ago. And she had been in type 1 diabetic for many years and was dealing with severe hypoglycemic events on a regular basis, to the extent where she was at the shopping mall with her young children, 3- and 5-year-old children, and passed out as a result of one of these events. And so was dealing with a relentless disease that had an impact on every single moment of her day, and now has been 7 years insulin independent as a result of an islet transplant, to the point that she now says she checks her blood glucose every couple of weeks just for fun to see that it looks normal. And so we're really motivated by this clinical proof of concept, and actually think it's an important de-risking component of what we're doing at Semma, because we know if we can get the right cells produced, and I'll show you some evidence that we can, and deliver them back to the patient, that we should be able to achieve this kind of meaningful clinical result for a much broader population. The two things that limit the applicability of this clinical islet transplant to date is the limited supply, as you can imagine, with organ donor material as the starting source, and the requirement for long-term immunosuppression, which has its own sets of morbidities and risks, which make the risk-benefit calculus a really difficult one for many patients. So our company has set out to solve both of those issues, and as I said, builds on really those two technologies on the cell side and on the immunoprotective bioengineering side. So the company was built on this technology, which is here depicted just as a cartoon diagram, starting from a cartoon red pluripotent stem cell. And this picture here is meant to simulate the type of cell fate choices that we now can control and master in a manufacturing process to take that pluripotent stem cell, which has the ability both to expand indefinitely, more or less, but also to turn into any kind of cell type in the body. And we can really control cell fate now at each of these fate decision points and turn those pluripotent stem cells into pancreatic islets, including those cells that make the insulin for the patient. And so that's depicted as this little purple sphere at the bottom. And then the second piece of this is, how do you deliver that back to the patient? And I'll tell you just a little bit about that in a few slides. So the first couple of slides, I'll focus on the cell side and try to convince you of what we've been able to generate in the laboratory. And this is a cross-section of manufactured stem cell-derived islets on the left-hand side, and a cross-section of natural pancreas-derived islets on the right. And it's a very high-level picture. You can see insulin expression in both cases, and expression of other beta cell genes, and we've done single-cell RNA sequencing and lots of molecular characterization, but the take-home message is that we really are now able to make something that, for all intents and purposes, looks just like a natural pancreatic islet. More importantly for us, as a cell therapy company, is how those islets actually perform. And I'll show you just one set of kind of potency data for us, which is a glucose-stimulated insulin secretion assay, where we're now able to produce in bioreactors at our clinical manufacturing scale and with clinical materials and GMP cell lines, stem cell-derived islets, or SC islets, that are as potent or more than those that we can isolate from an organ donor from a cadaveric pancreas. And you can see in this assay the cells are challenged either with low glucose, which is the blue bar, or high glucose, which is the orange bar. And what you see with normal islets is a stimulation of insulin secretion. There's very high variability in organ donor tissue, which is probably not surprising. But you can see in the center here our current production protocol, which we're bringing into the clinic at large scale, where we're able to see very highly potent cells in the dish. And so, of course, we focused on all of the aspects over the last year or two of bringing this manufacturing process to a state that it's ready to move into our first clinical trial. So focusing, again, as I alluded to, on the raw materials side, including, importantly for stem cell therapies, the starting cell line, focused on the composition and purity of this process, making sure there are no residual pluripotent stem cells, and potency, as I mentioned, as well as the manufacturing scale. So I'll just show you one picture of how those cells actually perform in some of our preclinical animal models. In this graph, you see a series of colored lines, and each of these colored lines is an individual diabetic mouse. So they're induced to become diabetic with a drug called STZ, and the axis here is the blood glucose level. So you can see we can make these animals extremely diabetic very quickly, and then implant them with our graft, which is those manufactured islets in the dish. And what's been really exciting to see is that those implanted cells start to control the blood sugar of these severely diabetic animals immediately, which is consistent with their mature, fully differentiated phenotype. And that ability to control glycemia in these animals lasts really for the duration of the lifetime of the animal. And you can see in this experiment out to about 200 and 250 days that if we take those cells out, whether at an intermediate time point or at a terminal time point, if we take those implanted cells out, the animals quickly become extremely diabetic, again, confirming that the implant of the cells is responsible for that efficacy phenotype. So, of course, in the last slide, I was showing you this in vivo data just with the naked cells themselves. And there is a path that you could take into the clinic with naked cells, really recapitulating the immunosuppression approach that's been done with cadaveric islets. But we're very interested in expanding the product to a larger pool of patients without immune suppression. And so we've been working on a couple of different approaches to encapsulating and protecting these cells. Here depicted in this cartoon is a physical barrier where the stem cell-derived islets are in the inside of a physical barrier, and that barrier prevents cell-cell contact with the immune system, but has pores sufficiently large to allow nutrients, oxygen, and insulin release in and out of that bioprotected membrane or biomaterial. We're also interested, as I said, in immunosuppression-based approaches and engineered cells, and have had discussions with a number of you on that type of approach. But I'll just show you, for the sake of time today, a couple of examples of the encapsulation approach that we've been working very hard on. This is a Semma's macroencapsulation device, and what's very exciting to us is this is the same kind of assay in a small rodent model, induced to be diabetic, and then implanted not just with the cells, but with the cells inside that immunoprotective encapsulation device. And you see really beautiful glycemic control and very, very high levels, actually, of secreted C-peptide in those animals. And we've also been collaborating with a couple of other scientists in this field, including Dr. Alice Tomei at the University of Miami, who've been developing what we think are pretty innovative alternative approaches to encapsulating the cells. In this case, she has a mechanism where she can take every individual islet and coat them with a biomaterial that protects them from the immune system. And so you essentially have lots of little islets that are free-floating but protected from immune destruction. And what you can see in her data here is the conformally coated, these encapsulated stem cell-derived islets we provide her are the purple line, perform as well as naked cadaveric islets in this diabetic animal model. And again, have taken it out to that point of retrieval and demonstration that it's responsible for the function. So we're really excited about the engineering side of what we do, and really devote about half of the company to new materials and new geometries, and have our favorite that we're planning to move into our first clinical trial. So speaking of clinical trials, just highlight at a very high level some of the other advantages I think we have in type 1 diabetes. I think one I mentioned already, this clinical proof of concept is already established, but also the early clinical trials require very few patients and have very clear, sensitive, and early endpoints. These are patients who are making really minuscule levels, if any, of insulin on their own. And so we have a perfect biomarker in a sense, because we can measure the insulin in the bloodstream of these patients after receiving our product and look right away to see if we're having a meaningful benefit for those patients, and really iterate from there. And we, of course, have brought in a team of people who have brought these kinds of cell therapies forward before, and are excited about what's happening with FDA and regenerative medicine advanced therapies as well. So I think we're very much set up for success in the sense that we are developing this off-the-shelf allogeneic product. We're in the process of developing manufacturing processes from the beginning that are very scalable and hopefully will be applicable to the millions of patients that could benefit in the future. And we're interested in leveraging what we've learned and what we've developed in terms of know-how, both on the cell biology side as well as on the device side, to move into additional both naked and encapsulated cell therapies in the future, which we'll expand on in the next couple of years. So I'll just end with a slide or two on our leadership team. So we have a new CEO joined us about 4 months ago, Bastiano Sano, who comes from Novartis and Magenta Therapeutics, has been working on developing cell therapies in a variety of spaces very successfully. We're really glad to have him join, as well as Dave DeGusto, who many of you may know, has joined as Chief Technical Officer, responsible for our manufacturing, and really couldn't ask for better experience in this very complicated cell therapy manufacturing space. And we'll have additional announcements of really exciting new people joining our team very soon. And I'll just end with our expanded board and group of investors, and we really couldn't do what we are doing and have done without them. And I'm really pleased to share that we've now raised over $150 million in capital, including a $110 million Series B late last year. And so brought on board additional investors, including Eight Roads and Cowen, and are really delighted that they share our vision and mission for curing diabetes. So thank you.